A very good morning to all the participants. Warm greetings from St. Pious the 10th Degree and PG College for Women, Hyderabad. We welcome you all for the international webinar on functional nanobiomaterials in biomedical field. We are fortunate to have an eminent speaker, Dr. Prabir K. Patra, with us, who is an enthusiastic professor, innovative researcher, and a motivator with an aim to inculcate scientific temper among the student community. He is the founding chair, Department of Biomedical Engineering at the University of Bridgeport. His research interests in multifunctional nanomaterials, polymeric nanocomposites, tissue engineering, polymeric nanocomposites, bioelectronics.
You have been muted, so please unmute. Your voice is muted. Please unmute. Yeah.
Sir is a founder of Department of Biomedical Engineering at the University of Bridgeport. His research interests in multifunctional nanomaterials, polymeric nanocomposites, tissue engineering, bioelectronics, flexible nanobioelectronics, polymer gels, and hybrid nanomaterials for sustainable energy. He has published about 50 research papers in peer-reviewed international journals and many conference proceeding articles in his area of expertise. He has chaired sessions at American Chemical Society and Material Research Society meetings. Sir has 1 US patent and 3 invention disclosures. He obtained his doctoral degree in material science from IIT Kharagpur. He also has 3 years of industrial research experience as a senior scientific officer. With this very brief introduction, I would like to invite our speaker to take over the session. I am glad to invite you, sir, once again.
Thank you, Dr. Padmalata. Thank you very much for inviting me. First of all, I'd like to thank all the people from St. Pius 10 College, PG College. It's a fantastic place. Before I put my talk onto the sharing screen, let me tell briefly that I have been fortunate that one of your students from microbiology, zoology, and chemistry, Sindhu, is our current student here, a master student. She is doing amazingly well. She first connected me to Dr. Grace Bina Paul, and we chatted. Then I requested Sindhu that provide me the connection of your biotechnology department. So she provided Dr. Padmalata's name. So she did a lot of background work. So I like to thank her and all the students, faculty, entire community, the people from industry. I like to wish you a very safe and healthy time at this very bad pandemic situation. I wish all of you stay well, safe, and healthy. With that note, I will first share the screen of my talk, and let me know if I'm being able to share properly, and then we'll start discussion. Can you see the topic?
Sir, screen is not visible. Yeah.
Let me go back because somehow I'm sharing that. It was like last time. So please bear with me if I'm not being able to. Do you see the screen? Do you see the screen now? Please let me know if you are seeing the screen, then I'll start. Please let me know. Please confirm me if you're seeing the screen, because I have no idea of checking whether you're seeing the screen or not.
Screen is visible.
Visible? Okay, great. So I'm going to talk today. Once again, thank you very much for inviting me. I'm going to talk in two parts. One is mainly for research, students, faculty, people from industry. You can all enjoy this. And second part is particularly geared towards prospective students who wants to come to United States and who wants to pursue the career in biomedical engineering, biotechnology, or associated health, pharmaceutical technology, that area. So my talk first is functional nano biomaterials in bioengineering. You all know, especially after this COVID-19 situation, the demand for biomedical devices and how those devices and technology controls drug discovery and design of medicine has been skyrocketed. So how important this field is, not necessary to say. Everybody knows here in this crowd. But let me tell you, my presentation outline today would be the nuggets of entire research that my research group does. We have a lot of students who are working in various areas of research. Then we have myriad of collaborators with whom we work. So I'm going to show you some of the area of bioengineering work and how important, how creative they could be, and how impactful they could be made. So I'll talk a little bit about tissue engineering. And this is not about just a discussion, it is about the work that we do in our lab. Then I'll talk about how these scaffolds helps growth of brain organoids and heart cells, skin cells, and various other cell types. Then I'll switch gear to discuss to you about biomimetic materials, which I mean is there are a lot of interesting things happens in nature, how we can copy those things and make those stuffs in the lab. So how we can mimetically design wonderful nanotechnology driven products and how that could be useful for health. Then I'll get to water tribology on graphene, how the water droplets or any other kind of liquids can pass through a surface. Or in other words, once that is known, you can design very nice self-lubricating surfaces for drug delivery. Then I talk about supercapacitor biosensing, how we make supercapacitor, how they store energy at the same time, how they could be useful for amazing biosensor for detecting or diagnosing molecules at a very fast rate. Then I'll talk briefly about brain organoids because we do a lot of work on brain to understand human cognition, to understand cognitive disorders, brain-related disease. So I'll talk a little bit about that, and you can see some of the amazing brain cells, how they are grown, how they are understood. They're very complex, but how engineering can help. Then I'll talk about fantastic nanomaterials, quantum dots, how they're being able to use on a surface to instruct DNA. So we can attach or detach DNA based on our own instructions by patterning this graphene quantum dot on surfaces, and hence you can make soft devices. And I'll also make an analogy as to how COVID-19 strains evolution could be understood and how fast they could be detected and even how they could be minimized. So there are some efforts we're doing towards that as well. Then I'll make another presentations on a career for prospective students. So why it is important for many people, at the same time important for me, why I was interested in doing something on scaffold. why tissue engineering was important to me. So there's one fact given in a journal many years ago, and that kind of triggered my interest, and I came to this field. And that facts are, as you can see, every year in the United States alone, 2.4 million burn injuries demand medical attention. 12,000 people die every year of burn-related infections. And if you do some associated diabetic foot ulcers treatment, that is $28,000 per patient. So this is a statistics given by Journal of Wound Care in 1992. So if you take another 28 years that has passed, and if you include all other countries in the world, these numbers would be staggeringly higher. So there is a need of grafts that could help to some extent, if not completely, for the burns or other injuries that happens to the body. So that kind of triggered my interest to come to this field. So to develop a scaffold where biological tissues will be grown, the very first thing is to develop an artificial extracellular matrix. ECM stands for extracellular matrix, which is our skin. Now, what skin consisted of, how their structures are, I'm not going to discuss because it is a simple biology. It has been known for years and years, but we'll focus on one certain thing, that is collagen fibers. So this is what our skin collagen looks like. They're very well aligned, bundled structure of 10 to hundreds of nanometers. So when you look at your body has nanostructures. And one of the prime examples is the collagen. So collagen that our skin has, they're actually tens to hundreds of nanometers, well aligned bundled structure. So any scaffold that you want to make for growing tissues, that has to be first, can we make these in the lab? Can we mimic the scaffold that is exactly similar structure? So again, remember, many of you are student here. When you do something amazing or extraordinary or creative, you don't have to be very superfluous. You can look at the nature. And the very onset for bioengineering or health is look at your body. That is one of the best machines, or combination of machines, if you will. So this is one collagen that our body creates. Very nice, fine nanoscale structures. We have many other nanostructures in the body, but we're talking about collagen right here. Now, how can you make it? So go back to history. The textile engineers or textile technologists, over the years, they have used various techniques, blow molding, extrusion, calendering, weaving, various other things. But they use something called, blow molding, where there are little orifices, very small holes. They'll put polymer melt, and then under pressure, they'll get the film out from the other side of the orifice. And by that method, they used to make, films or fibers as small as microns. Unfortunately, that doesn't work in this case. So we have to find a way. So there's a method named electrospinning. So electrospinning, I'll go slow and I'll explain to you. And later we'll take questions and we'll go in details if you have specific questions. The electrospinning is a process which is not new either. It's a 100 years old method. But the idea is, can we use it new? Can we design it in a way that can serve new purpose or modern day needs? For example, having scaffolds. So there are various groups have been working on tissue engineering in this country and in the world. So we started. Electrospinning is a process where you use a syringe. In that syringe pump, you have a needle. And in the needle, you can create your own polymer solutions that will be the scaffold for the cells. And those polymers could be FDA approved, biodegradable, biocompatible polymers. Food and Drug Administration, FDA. Their approval is necessary for dealing with these polymers so that cells and tissues that grow, they don't die on this substrate. So electrospinning process is this. You have a syringe, you have a polymer solution. That syringe, when it is under electric field, so this plunger goes forward, the liquid is ejected from the nozzle, and then it forms a unstable structure and then gets collected onto a collector. And those collections of these are at a very, very fine scale. So left side is a schematic, right side is one simple apparatus that we used. Then we modified those apparatus. So what I'll do is I'll first tell you very briefly about this process. When something we start with a millimeter nozzles, and that produces 100 nanometer in size. So you can imagine how many orders of magnitude we are reducing the size scale. That's a lot. And for that to happen, you have to understand the physics. So there's a lot of instability physics involved. We did this with various groups while I was working with UMass Dartmouth and in collaboration with MIT. We did a lot of work on that, and we explained the reasons behind as to why this nanostructure forms from millimeter. So that has been detailed by us and other people elsewhere. So here I'm going to show you a simple figures where we have shown that when you collect these small fibers like collagen, excuse me, they're artificial and they're very small in size. So you can change the size of these by various design of electric field. So if you look at the left side, it has a needle, and needle is passing through a ring, and the ring is attached with a power supply. So here needle is not directly being charged, but it is induced. So it's called inductive charging. On the other hand, the right side, if you look at, needle is directly in touch with the charge. So by various charging scheme, you can make small fibers, you can make intermediate size fibers, you can make larger fibers within the scale of 10 through 500 nanometers, if you will. And we were the very first, very early, who determined the charge on single fiber. We're talking about a fiber that is 10 to 100 nanometers in diameter. And we're talking about a charge. Charge meaning when you're touching the needle, the tremendous amount of charge is being given to this liquid. So when they stretch to a jet formation, there are a lot of charge into it. So how can you measure that charge so fast? Because this charge relaxes. The reason for having an importance of measuring this charge is when the fibers are collected, if they still have charge for a long time, then when next batch of the fibers come, they will repel and some of the fibers will be floating in the air. We don't want that. If that happens, then there'll be a lot of porosity and fiber diameter changes. We need to measure the charge, and hence we can measure how fast they can relax this charge. So we calculated by our own design. We have Faraday cup, we had nanocoulomb mirror, and we connected those to the fiber, and we measured the charge as 35 nanocoulomb per milligram. So think about this. A single 100 nanometers collagen fiber-like fiber has charge 35 nanocoulomb per milligram. So measuring the fiber charge of that small density itself is a feat. So it was very early. So this work was represented very well in the community. So now I'll go quickly as to why it's not moving the slides. Yeah. So let's see. Okay. So we measured the charge, and then we thought these fibers that we collect, they are not aligned. They're random. But the picture that I showed you on our screen, they're aligned. So one of our students came from Bombay. He developed a device, and look, let me tell you, that is what called creativity. This guy is now working for Illumina, one of the very big company many of you know for DNA sequencing or single cell sequencing. Illumina is one of the largest sequencing apparatus manufacturing company. So what he did is he's a very good engineer. So he took a scrap computers, and we gave him some supports to build the device, and he made a rotating collector with a speed, with switch, with all the programming put in, all the coding, and then we started making a line. This paper has been detailed, published somewhere else. He did mathematical modeling also. He's an amazing guy, Kapil. His name is Kapil Mahakalkar. He's from Bombay. So with that, we made line fibers. You can see some of the real image of these aligned fibers. So now I'll quickly pass through. We made various type of these structures. Some are random, some are aligned, some has beads on them, some are little flat, some has vesicular-like structures, just to optimize what is the best for these cells to grow on. And I'll show you how cells grow. So we determine various properties. I'll not stay details on those. Viscosity was changed, surface tension was played with, field strength, meaning how far distance of the collectors from the needle tip is, and how can you calculate those various parameters roll into forming a good fiber. With that, somehow, sometimes this is being frozen. So let me take a look. Sound system, maybe from my side, is not is interrupting a lot. I'm not being able to move the slide. Bear with me. Somehow something is, yeah. So now we did various analysis on this. Again, it got stuck. I apologize. Somehow, maybe from my side, link is not happening properly because it is just always going back. Okay, so we played with all the parameters, and you can see this is where we started growing our own cells on these scaffolds. So this is a real skin cells that we got from NIH, fibroblast 3T3 skin cells. You can see day 1, day 3, and day 5, how they covered within 5 days. They covered the entire matrix. So this is the real cells you were seeing. We did a lot of analysis on how these cells basically grow. We did histology of various proteins, and we saw from bottom to the top, they're covering the entire scaffold side. So as you can see now, to take a look at this picture, and later on you can think about it because I'll discuss about this. Okay. So then we did important protein, fibronectin. So when cell grows on any scaffold, cell secretes some of the proteins from the serum, secretin. And then there are proteins that help them bind onto the surface called adenosine. So these adenosine and secretin, they talk and they bring more and more cells, proteins like that, form a pathway, more and more cells come and they grow into tissue form. We studied all these proteins and we figured out that there is a diameter which is 428 nanometers that is optimized for these cells to grow on them. We also did with type 1 collagen and got the same results. And remember, when skin formed, the very important aspect of this F-actin organization. So I'm not going to talk much details on this. Later on, I can take questions from you. F-actin is a protein whose organization onto the surface is very important for any cells to grow. In this particular case, skin cell. So we saw how these F-actin is organized from bottom to the top with a given serum concentration. And that is exactly like our body has. So you see left side serum concentration 0 to 10%, right side is the F-actin morphology and their stress distribution. If you look at the bottom part, right where my arrow is, that's where it is formed complete actin-like morphology, ready for the cells to grow into tissue. So we can say safely that our scaffold is working very well for the cells to grow. We also saw the role of specific surface area over volume. As the fibers are small in size, this specific surface area plays a very important role for the cells to grow and form tissues. So you can tell, smaller the fibers, better it is for the cells to grow. So on this, I'll go quickly a summary on this particular observation. In this case, we used an FDA-approved polymers called PCl, polycaprolactam. That is our scaffold. We used skin cells to grow on it. And we found out by playing with all the parameters and using electrospinning process, we can form a scaffold that at 400 nanometers and smaller, it can grow skin cells very well. They grow, they attach, they proliferate, and they form into tissues. Now, I'll take that, okay, it happened for the skin cells. How about heart cells? So that, many of you are aware that heart disease kills most of the people in the world. So they're the number one killer disease is the heart disease, and distant second is cancer. So that is the statistics. I feel like it's important for you to understand and for me to share with all of you that heart disease is very, very important to, all diseases are important to cure and solve, but people have misconception as if cancer kills more people in the world. That is not the case. It is the heart disease. So we started studying cardiomyocyte cells, which is the heart cells, on these scaffolds, and we saw very nice sarcomeres are formed. Wonderful heart cells follow a particular scaffold that is electrical gradient. So heart cell is different than the skin cell. So you have to do something onto the surface of electrospun fiber that we made. So what we did is instead we made a combination of PCl and graphene. So graphene is conductive, PCl is not. So it has a conductive, non-conductive, conductive, non-conductive interfaces that creates electrical potential. Somewhere conductivity is high, somewhere low, somewhere medium, somewhere low, like that. So when you form these, connections, myocardium contractile forces, just the way heart does, it happens through electroconduit. And I'll show you how we were able to create an artificial heart-like structure where actually the heart cells grew. So this is here I'm going to show you a movie, and this movie is about on our scaffold how heart cells are beating, and that beating is as good as human heart beating. So you can see that here. So let me play the movie for you. You're seeing the movie, right? You see that is our heart. So this is a heart grown on a scaffold in a lab. It is not the human heart in vivo that you see. So what does it tell? So we measured the frequency of beating. Frequency is 1.5 to 0.5 to 1.5. So our human hearts, they beat 40 to 120 beats per minute. And that beats per minute, if you equate to the frequency, they are 0.5 to 1.5 hertz. And these heart cells that beats onto the artificial scaffold, or the heart cells that we produced into tissue form, heart tissues, they're exactly that beating. So there is a hope that we can study some of the heart disease and heart aspect also outside on a scaffold without getting into human body and understand it first much more depth. So then we took a step further. We thought, let's do something biomimetic. Biological tissues have the remarkable ability to remodel and repair in response to mechanical stress. So we're trying to understand, can we really make materials that behaves like tissues and not tissues? Or they can adapt to any force or any weight are given upon them. They can adapt to that. So when people go to gym and do exercise, you see what happens to the muscle is there are fibrils in the muscles, and then there are a lot of muscles, they get tear, the fibrils, and they form loop. So when you put more and more compression onto your arm, the loops expands this way, and these are aligned that way, so you get stiffened. There's no extra muscle coming in. It is basically forming a very nice loop structure. So various such structure you can form and understand those biological processes. So we did some of the study. Um, we made an artificial material that behaves like cells. So I'll say carbon nanotubes and PDMS, polydimethyl cellulose. I'll not stay much more details on this. I'll pass by. I'll tell into a storytelling form. Then when students have much more specific questions, we'll be able to see. The goal was, can we really make an artificial muscle-like actuator? But they are not muscles, but they behave like. So we use this hypothesis that compression induces alignment of polymer network ends, and that continues to have increased stiffness, and you can form a structure. So what we did is we formed a vertically aligned carbon nanotubes by a method known as CVD, chemical vapor deposition. And on those aligned carbon nanotube, they're very small, 10 nanometers in diameter, and equidistant they're apart. We put a polymer, oligomeric form, and then polymerized it, and we formed PDMS CNT structure. Then it took the catalyst away and formed an open-ended composite, as you see on the coin. That black, blob that you see, that's our composite. It has a unique behavior. So we started giving a dynamic compression, and we saw these are published in details, as you can see. If you want to know more details, these publications are there. Anybody can look up. But to cut the long story short, these composites are such, more and more compression you want to give, stronger it gets. The more stiffening happens onto the structure. We call dynamic strain hardening. With a very less strain, they get stronger as you go by. And you see, they're going to have their stiffness up and up and up after many cycles. Then we thought, okay, that is an artificial materials. Can we make something that our body has similar behavior? And we went to liquid crystal elastomer. Now think about that. I am presenting bioengineering. I'm not only presenting biology here. Our body functions in a way where they use engineering principles very detailed. So to tackle certain problem, you have to have an interface between engineering, medicine, and biology. And that is actually essentially a bioengineering program, which after COVID-19 And as we enter into infectious era, many of you have read Nature magazine. They have defined the future times that we're going to go through now is infectious era. A lot of infectious disease will come up. Why? Because there are a lot of animal-borne pathogens. They'll be creeping into human society. Our immunity is not ready for that. So a lot of tussles between those pathogens and our body structure will happen. Therefore, it is important to develop biomedical devices, to do a lot of computational bioinformatics to understand the strains. And based on strains evolutions, you can find active sites. You can determine molecules that can bind to those active sites, and those molecules are drugs or vaccines. So there are tremendous interdisciplinary effort must be put forward to tackle this infectious era. So in this particular presentation, as you were seeing, these LC liquid crystal elastomers our body has. Our body has a lot of liquid crystal elastomers. For example, various lipids or lipid bilayers, if you talk about phospholipids, they are liquid crystal elastomers. So when something is compressed, what happens to their structure? So we wanted to study those and biomimetically come up with a way that in future, if anything happens, we can basically address some of the needs. So first one was artificially synthesized. This is liquid crystal elastomers, and we started seeing that if we continue to increase the cross-linking content, liquid crystal content, and we have a mesogen meaning the molecules that under compression can order in a particular direction. If we play around with these parameters, you can form amazing materials, which is the white stuffs right here. This is called liquid crystal elastomers. Our body has this stuff. And we saw they get stiffened. That means, once you understand these, you can understand how our body tissues can adapt and remodel themselves to the stress. So stress markers could be related to these issues, and you can say what kind of health concerns might come if they don't respond to this very well. So that study we did, we published in Nature and various other great places, and they could be discussed further. So we did those. So you can see, as you can see, the time passes by with cross-linking density and liquid crystal elastomer content, their stiffness goes up. I'll not go much into details. We did analysis with 2D wide-angle X-ray diffractions, we did with polarized light microscope, and from there we show that when you compress dynamically, remember the word is dynamic compression, not static. So when dynamically compress these kind of structures, what happens is the various mesogen units gets away from the compression axis and they align mostly into XY plane. Then we calculate the order parameters, like how these molecules are getting ordered. So the order parameters S is calculated by making 3/2 cos theta plus half. Theta is this axis where you are giving your dynamic compression and from where these mesogen units are getting aligned. So that angle is theta. And we can show from all these images, both X-ray and polarized microscope, that orientation factors of these LCs are highest in XY plane. So we moved further, and then we thought, okay, we have artificial tissues, we have scaffolds where we grew heart cells, skin cells, and various other cells. Now, can we make a surface where we can deliver the drug very well? So here I'm presenting to you the deep science behind it. Not exactly drug delivery device per se, that is an ongoing work. So I'll give a quote here. God made the bulk. Surfaces were invented by the devil. That is Wolfgang Pauli's comment, meaning when you look at the surface, they're very complex. You have to understand surface very well. That's why we use the word surface tuning, surface tailoring, surface engineering. Those are the words comes up. What it tells us that when you slice any bulk into surfaces, you're basically slicing into nanoscale. When things become nano, things get complex. So you have to understand much more detail. So when you rub between surfaces, you can have a debris, you can have plowing, you can have tribofilm, you can have adhesion. The various things happen, but all at a small scale. So combining Amonton's law of physics, that when you apply a force on any surface, how much force you apply is proportional to the area on which force is applied. So that's the friction. In a classical mechanics, friction is when two surfaces rub against each other. The molecular deformation or reorientation, that's the friction. So combining these two, we came up with a very unique surface we created ourselves, we thought that can continue to reduce the friction. Or in other words, the lowest frictional surface can we really create. Snail creates its friction by its own engineering. Do you even know how snail moves? Snail's locomotion, move is locomotion. It's like an engine. So what it does is it throws mucus in front of it. Then it puts its body mass on the mucus. So under shear, the mucus thickens and becomes very icy layer, slippery, and the body slips through. Then the moment weights is lift up, this mucus becomes thin again. I mean the viscous again, and they throw it forward. That's how it moves. So amazing of these surface engineering that these species do. A human being have a lot of things to learn from and re-engineer those structures. So we made one such, and we call it centrifugal adhesion balance of graphene. We call CAB. It is not the cab that you ride from place to place, but it is a cab where molecules ride. So in this case, water molecules rides on graphene, and we see how graphene behave under water molecules. So this is what water happens. If I ask anybody in the crowd later on, you all will know that graphene is hydrophobic. It doesn't like water. But graphene exactly is not hydrophobic. It's pseudo hydrophobic. Or in other words, any surface, mica, sand, polymers, cellulose, metals, you name it. When you apply force and then slide a drop of either polymer solution or liquid, the way force and time curve varies, your force goes up and then it plateaued. So force over time, force is in y-axis, time is in x-axis. So you can see they go up and then plateau. The reason this work was accepted again in Nature is because we first showed that in this particular case, it doesn't follow those curves. The time at which water molecule drops is invariant of the force it takes, invariant of time. It goes very fast. In other surfaces, it takes 8 to 10 minutes to have the curve stabilize. In this case, it's in few seconds. And there is no upward trend to plateau it. It just goes parallel to the x-axis. So we thought that means what we're essentially creating is a very nice surface where graphene molecules are not getting a chance to reorient or intermolecular change of that. Hence, we are creating a very low lubrication surface. Excuse me. So now you can make, let's say, you can anchor certain molecules on graphene, and then put the water droplets. It might not spread that fast, but slow. You're creating a gradient-based friction on a surface. So when drug is delivered through either any capsules or in nanoparticles or via any delivery device, it has to go through a tortuous path. But if you have a less friction surface, it can go fast or go slow. You can control that. So delivery design is very important. And from that work, we got a lot of inspiration. This work was directly not bioengineering, but it kind of paved the way for some great ideas of doing delivery device, drug delivery, if you will. So this is one of the interesting work. Then we thought, can we make 3D structures of these? So here we made a very nice fabrications using mechanical engineering and some aspects of chemistry, and then we formed 3D graphene oxide solid. And if you look at later, graphene oxide was reduced into graphene. Now we form a very nice conductive porous scaffolds as if they are stacked. Now inside those scaffolds, I can put some brain organoids. Like we can grow some brain cells, and both sides we can put contacts and connect to the computer device. And you can see how brain markers are sending signals all across. Then map those signals and understand what is happening in the brain. So see, these same cascades are now a device that can probe questions to the brain. We all human beings can talk, but cells cannot. But if you can give some probing, cell gives out some response. Then we develop logic chip. We took microcontroller, we took Arduino board, then connected all these, and then found out a conversion between digital to analog. So when that conversion happened, we were being able to translate questions to the brain. The brain gives out signal, and those signals are digitized now. How much voltage, how much current, what is happening with the cell stress. All this status we are knowing from the numbers. So it's a great piece of studies that we ended up doing. So I'll show you those brain organoids and their structure in a subsequent later slides. But let me just show you, here is the work where we made supercapacitors. So think about how connective this field is. When you say sensors, we talk about electronics, detection device. But when you talk about supercapacitors, the very first thing come to your mind is it's a capacitor. So it's storage energy. It stores energy and keeps it. It doesn't discharge very fast. At the same time, it charges pretty fast. So supercapacitors are devices that follows Faradaic and non-Faradaic charging mechanism. So what we prepared is we prepared a very nice nanocomposite system made out of carbon nanotubes, graphene, and cellulose paper. And then you can see here we're peeling it off and then made a very nice structural analysis. And you can see 20, 14 nanometer scale how layered these structures are. The top, a little bit higher resolution, bottom, very lower resolution. Here you can see 5 microns, but you see the layers. And each layers are about 20 nanometers separated. It's a very nice layered structure we created. This is microscope. So we have to believe that structures are indeed true. And then what we did is in a supercapacitor device, you need electrodes, you need a separator, and then you have contact electrodes, right? So the separator also we made ourselves. We didn't take commercially available cell guard, but compared with it. Normally what you do is we take our aligned fibers, as you see in the middle picture, and then wrap some filter paper with it, and soak it into sodium chloride and put into as a membrane separators in the supercapacitor device. I'll show you how our supercapacitor device was built. So you can see here metal cap, ring. So you have to have engineering skills. You have to build stuffs, and this is more important at this time that we have to build things. People are building masks, people are building ventilators. So you have to make those. That's why you are engineers or biotechnologists, not just only drug discovery or drug design, but the other side. So we made these supercapacitors ourselves. So this is our own supercapacitors, and if you look at this is the capacitors. So how good these materials are would be determined where they exist in Raygon plot. So there are a lot of understanding of supercapacitor physics. I'm not going to discuss those, but Raygon plot and Bode plot, they can tell this your supercapacitors is exactly that square by measuring specific capacitance, by measuring charging time, discharging time, and current capacity, current density of these. So we measured all, and we saw there at the very top. So this is one of my PhD graduate students' work. So what he did is he made our supercapacitors and took a battery commercially available of same nature, but ours has only 2 coins here. 3 capacitors that we made, supercapacitors, if you will, and we put into series, and you can see they're producing same numbers in terms of current. But this is way bigger in size, and this is just little. So this nanoscale structures, if you do very well, it does amazing, and then it does amazing stuff. Then we proved that our supercapacitor is really working well by connecting them in a series and lighting up a 2.2 volt LED. So we actually built the supercapacitor that is in working conditions, as you can see. And Assis, as a PhD graduate student, he has done postdocs. He's now working. So he published it again in Nature Scientific Report. So it's a very. Then we use the same supercapacitor device and now started sensing biological molecules. In this case, we can make graphene, polypyrrole, and then have avidine. So avidine binds with graphene. And on the top of the avidine, we come up with a very small concentration of various types of DNA and see how small of DNA dose we can detect with this device using supercapacitors. We are consistently detecting micromolar quantity of DNA solutions, DNA structures, and then we're getting into nanomolar quantity, where we have been very successful. Again, this is another graduate student of mine who developed his PhD thesis on this and published these works much more details. He came up with this concept. And then we did computational biology and bioinformatics. In this presentation, I'm not talking about computational biology much, but we have tremendous amount of efforts in our lab doing bioinformatics and computational biology, how various sites of these proteins are binding onto our graphene sensors, and how we can say the structure of protein folding or changing. This is very important at this time, particularly because there are various computer science groups, those who does computational biology and bioinformatics, and we do some part of that work as well, are working around the clock to show the variation of this COVID-19 strain. COVID-19 is a virus that has a strain hiding behind its own hydrogel. and that strain is the most dangerous strain. So how that strain evolves? Protein changes its structure in femtosecond. So if we do folding study very well, we know site S1 goes to site S2, goes to site S3, goes to site S4, how they change the structure. And if we know the structural change pattern, we know the active site's nature. Then we can find out theoretical molecules who will bind to those sites and make them functionally weaker, if not non-functional completely. Now, those molecules will be now for pharmacists to, or pharmaceutical engineers, or pharmaceutical technologists, or scientists to develop the molecules as a drug component. So there are various companies like Moderna, CureVac, or Remdesivir, and then you say Pfizer and Novartis. The way you see they're working exactly like that. So there's a very nice link between electronic sensing, computational bioinformatics, and making the real vaccines. So these are very interdisciplinary and very, depending on one another. So we do a lot of such computational work in our lab as well. This is the time. And you see, this is the sensor device actually. This is the micro electrode on which you put this graphene and CNT and avidin, and then you're putting DNA into it. For that matter, any strain we can determine. So this is the supercapacitive biosensing that we call, but using a microelectrode. Okay, here is a work making 3D structure using bacteria. So bacteria is a species to biologists. Bacteria is truly an engineer to engineers. Bacteria can help making 2D to 3D structure. Bacteria can help making a logic circuits language in a board. So with these bacteria, we moved electronics. These bacteria have very nice crystals of magnetite. So they have iron oxides in them. From the media, they form iron crystals and make them aligned. And they move under magnetic field. When they move, these crystals, they orient themselves to the direction of the magnetic field. And very amazing images you can see. They're making 3D electronics, and at the same time, they're making their own crystals. If you look at these crystals are made by the bacteria inside, and they're amazing nanocrystals of iron oxide. If you take those crystals out, you can use them in hyperthermal cancer. So see, the bacterial species, they're navigating electronics and dropping it on a particular place. They're also helping us to write logic circuits chip and its language, and also making nanoparticles for cancer treatment. So it's a very multifunctional materials, if you will. And these bacteria is called magnetotactic bacteria. They're very safe, they're not harmful, but still we grow them in a biosafety level 3. That's the criteria we do. But it's a very fantastic work of truly interdisciplinary bioengineering. Now, graphene quantum dot. Quantum dots, they're so small, they are within the range of 8 to 10 nanometers, 4 through 7 nanometers, and 1 through 4 nanometers. By various techniques, we make them. And you can see all of these quantum dots from this table, they emit color. So they fluoresce. And those colors are yellow to orange to green to blue. That means you can use them as optical sensors to detect biological molecules. Or you can attach things on them, and from the fluorescence, you can tell what are the attachment kinetics or types. So they're used in cell biology very evidently. So we have a lot of collaboration going on this particular project with UConn and also UC Berkeley. So I'll show you a very interesting work later on with these scales. Now, if you recall, I talked to you or promised you that I'll show you some brain organoids. We're doing interesting work on 3D bioprinting. We call it bioprinting. So, in brain, a very interesting area, but very less chattered territory, or in other words, very less explored work is the spots where various organoids meet. So, for example, our brain has cortex, our brain has thalamus. So you can create cortical organoids, you can create thalamic organoids, but when they fuse, those spaces are aerial spaces. Those spaces are not explored very well. So you have to get in details to explore those. So here, with a simple technique, what we did is we did human embryonic stem cells. From there, with stem cell treatment technique, we made human cortical organoids. Then we also took human thalamic organoids from stem cells, and then we fused them with the bioprinting, 3D printing. And when we fused, we got a new lineage. This lineage basically cortico-thalamic organoids. For the first time, we reported that this lineage If you understand well, you can talk about axon targeting and reprogramming of the cell. But in other words, if cognition results are there, you can at least imply some of the pathway, not solve it completely, but that implication is very huge. Right side, you can see those brain organoids grow. So we grew in the lab 30 days, 60 days, 90 days. These are unlike skin cell and heart cell. They take many, many days to grow completely. Now we found something very interesting, I'll tell you, and that might alarm you a little bit. We use cell phone 24/7, and many of you use cell phone all the time. What if a pregnant woman, she has been using the cell phone 10 years prior, and then even during pregnancy and afterwards? We try to ask question to ourselves. Let us see, do these devices pose any problem to the health? It may not, but do they pose any problem? You might ask, radio frequencies, they're everywhere, Dr. Patter, in the earth. They don't pose any problem. They don't, because they are very well dispersed. When nature does a thing, it has evolved, and nature does things very well. We human won't do things that well. So we take years to perfect that. So my point of saying is that to mimic nature is possible, but nature has gotten years and years to evolve the technology, and we don't. Our lifespan is not that much. So you have to be very creative. So what we did is we grew these cells, the brain organoids, these are brain organoids, not brain per se, but brain organoids, very early stage of the brain. And we put electronic circuit beneath it. We also give radio frequency onto it, and we wanted to see what happens to this brain. So we saw that 30 days to 60 days period, there are holes in these organoids. Then when you pass 90 days, all holes made up, and then you see under RF and without RF, you don't see much of a change in 90 days. But in intermediate stage, the change happened. So we were alarmed that this change happened, but in 90 days we don't see. But there might have happened something from the marker point of view. Some gene markers might have been altered. So we tried to study all those, and we went to much further. And you can see this work is published in Cell Stem Cell from Cell Press. And then we moved, and we wanted to see the structure. We saw two things. One is the brain organoids that was under radio frequency, or let's say antenna effect, which were having holes in their structures, they indeed had some markers suppressed or lowly expressed compared to a normal healthy brain markers. So we can see that we're getting into a real curious world, that what exactly do they do. So that work is still ongoing, but we can definitely say that this cell phone, if pregnant women keeps it for every day in and out, who knows, the baby that is coming out might have some cognitive issues at the early. We don't know for sure yet, but we have seen clue that clues doesn't tell a good story. It might have effect. So all the cognition, all the disorders in the brain that you see of the kids, it may not be always natural, it may be our doing. So this kind of research will be very impactful for solving those diseases in future, where engineering is key. So but we also got some very good benefit. We saw benefit in the sense, by doing a good setup in my lab, we have been able to measure that we are creating blood-brain barrier artificially, but in these brain organoids. Remember, brain organoids is not a brain. Many such organoids forming the fusion is the brain. So very early, if we can find out inroads of the problems, you can solve. So here is a typical image of typical marker containing brain organoids, human cortical organoids, and this is vascularized human cortical organoids, meaning we form the vascular structure. We proved with a simple technique. It's called, many of you know, if there are any electrical engineers in the crowd or any faculty, you know that impedance, which is a change of conductivity as a function of frequency. So impedance measurement we did onto these organoids. We put the probe directly into the brain organoid. And we saw, in case of vascularized one, vascularization meaning more and more epithelial cells started coming in, epithelial cells started coming in, more and more cells come, more insulative the network gets. That means conductivity goes down. So we can pattern, and we can see the vessels or vascularized structures are forming. So it's a very early work of vascular structure formation in the brain. That's why we call engineering vascularized brain organoids. We're probably very early. That's why nature accepted this work. And many of you know that Nature magazine is very tough to publish. It's one of the very top journal. So then we calculated the Western percentage, we calculated the brain, organoid size, and we did all these studies. And you can see we picked up the right markers and see CD31, DAPI, how they are expressed. And we tried to propose that indeed we have been able to form vascular structure. We've been able to see the effect of some of these, outside field, whether electric field, electromagnetic field, or whatnot. That work, that field effect detail, that is still going on. I'm not reporting those yet, unless I'm very confirmed. So let me take step now. I spoke to you about cancer and heart cells. So once I talk about heart cells, I must say something about cancer. And this is an interesting work I like to present because this work was done by a high school student in my lab. So my lab has a program called No Fear, Nanotechnology Opportunities. No Fear is freshman engineers in advanced research. So I use freshman students onwards and high school students also in our research. So I gave her a project that you do computational work. Simply by doing computational bioinformatics work, can you tell how early extracellular matrix behaves when cancer cell invades? And can we really make a case that we can quantify that if this is the structure VCM happens, we can tell the cancer is spreading or not. So this is a completely computational work. So don't get me wrong, there's no experimental verification yet on this, but something good clue we are getting. And hopefully experiments will support that. This is called extracellular matrix manipulation to control cancer invasion. So here what we did is, if you look at the ECM matrix here, it's very random. If you look at the second one, it's very wavy, and these are very aligned, and these are very loopy structure. So in each case, you can see here the cancer, the black spots, that's expanding a lot, but left and right are not. And then we did various CompuCell 3D MATLAB programming, and then we use Hamiltonian functions, recode the equations, and put it in, and then we calculate each pixels under these invaded surface and calculated the pixel density. From there, the fiber distance, fiber angle, fiber density, fiber positioning, all we calculated towards predicting the ECM structure. So we meant if there's a wavy structure, cancer cells might prevent more. If there is an entangled structure, it might prevent more. So meaning soft and rigid structure, what role it has onto cancer invasion, we kind of gave a proposal for that. So that's why it was published in IEEE SCM Transactions on Computational Biology. So this work was overseen by a master's and a PhD student, but it was basically led by a high school student who is now in Carnegie Mellon University as a first year or going to computer science undergraduate student. So if you recall, I talked about quantum dot. So here you think about, we made 3 graphene quantum dots. We wanted to control how fast or how slow DNA could be attached on these surfaces. Or in other words, we can create a DNA template based on the instruction of the surface that gives to DNA, and we calculate all the binding energies or debinding energies, detachment energies. If you see, if you have a quantum dot that doesn't have any oxygen, that binds with DNA very strongly. You can see all the binding sites. If it has intermediate oxygen, low oxidations, very less oxygen, it binds, but not as strong it was before. If it has medium to high oxidations, it doesn't bind. The DNA goes out. So we are creating a surface that is very tailored, very engineered to attach DNA on it. Or in other words, we can template our DNA as per the pattern of the surface. This is a very good work with a group of Berkeley researchers, UC Berkeley researchers, and we then proved experimentally by X-ray photoelectron spectroscopy and luminescence spectra how this DNA is being controlled by the surfaces. And for that, we were able to create DNA template or a particular surface that can anchor to specific protein at one wheel. That's what we're getting at. Or in other words, nanofabrication of soft devices. So we did various other piece of work. I will go quickly on this. So research part, I will end right here. And if you want me to go to the next talk, like quickly about the graduate students' future, I will start the presentations, or I'll take questions first, answer your questions, and go back to the next, whichever way you prefer.
So I request you to continue with the career guidance at the
Okay,
end of the career, then we can go for question answer.
fantastic. So, do you see the screen now?
Yes, sir.
Okay, good. So let me quickly go through this. So this talk is about biomedical engineering, academic and career opportunities in the United States. So I'll tell you what is this field, how this field is growing, what are the job opportunities, and what it is going to be in future. My very fast answer, as you know, what it is going to come. It is one of the hottest field right now. Right now. So I'll show you what is biomedical engineering. Many of you probably have confusion. What is this field? It is a field that applied engineering principles in biology and medicine and improves human health through cross-disciplinary activities that integrate the engineering sciences with biomedical science and clinical. If you look at the biomedical engineering, application of principles of science and engineering at the interface between medicine and biology. So that's the biomedical engineering. For example, without that, the medicine development, medical device development, understanding genome, or for that matter, understanding underlying biological or biomolecular pathways to make devices for disease is impossible. So this is one of the very interdisciplinary field. So where do biomedical engineers work? Biomedical engineers, and this is from my experience, I'm telling you. So every year I go to Biomedical Engineering Society. We have a society. We have council of chairs. All the department heads, they meet. So biomedical students, they go to universities to do their PhDs, postdocs, or being faculty. That is one side. And it doesn't have to be everybody interested to be academia. You can work in industry, you can work in hospitals, you can work in research facilities, in academia and government. So you have all across the opportunities. I'll be more specific as to where our students have gone, so that many of the students who are here in biotechnology can form a great career path in this field. So this is a report given by Bureau of Labor Statistics. This is a report given in 2011. If you look at in 2011, the growth rate of biomedical engineering field is perceived to be 72% in BME position. Every year, 12,000 new jobs. This information is in line with similar report from a society called BOL in 2009. And the average salary, I'm talking about average, of a typical undergraduate BME student is close to $80,000 to $87,000. This was a report in 2011. This has increased now. Growth rate of women participating in BME is staggeringly the highest. So all the female students who are here in this presentation today, you should understand that largest number of female representatives are there in biomedical engineering field. And they are amazingly placed, and many of the women are world leaders today in this field. And I'll show you some specific example as to how this job growth rate is improving. You can see here, by 2024, you are still projecting to go up. The total jobs are going to be only going up. Now, with the pathogens and new infectious era that is coming up for the next few decades or so, I believe. Now, the medical devices, the drug discovery, computational informatics to understand, pathogens, and then drug screening, and then pharmaceutical development of those drugs, pharmacies, all together the allied health, they'll be the mostly funded field in this country as well as should be in the world. And in USA for sure, National Institute of Health has given the highest amount of money of this $1 trillion that it had. Out of them, half of that is gone to National Institute of Infectious Disease and Allergy. That has been given highest. So National Institute of Health is given highest money, meaning that field will grow in next decade that much. So the demand for medical devices, demand for devices that detects and senses, demands for devices that can also cure, or the medicines that cures, is going to be are going to be in very, very skyrocketedly high. So it goes without saying that this field is very important. And you can see total health area, biomedical engineers, home health aides, personal home care aides, medical scientists, physician scientists, skincare specialists, biochemists, athletic trainers, etc. All together, more than 65% of the funding would be given to this field. So no matter whatever undergraduates you have, if you come to do masters and PhD in biomedical engineering, one thing I can tell you that you have tremendous opportunities to get a job immediately. Many of our students who just graduated, one or two of them got 3 offers. So I'll tell you specifically those students, but they took, clinical engineers position. That is much more. One of my students directly working with COVID patients in the lab. And I'll give you an example. You should be excited. You know, you are reading a lot of things, right? This is also a time called infodemic, information pandemic. A lot of disinformation is passing around throughout the social media. Some of them are solid info, some of them are very misinformed. So this is a flu. This is a super flu. That's for sure. But the way this flu works, it takes the lung because lung has this alveolar sacs. And as I mentioned to you a few slides ago, that COVID-19 strains are essentially a strain that is hiding in a hydrogel. So when the hydrogel gets stuck into the alveolar sacs, the very first thing it does is it disrupts the communication between lung cells. But lung cells also have its own immunity. They try to fight it. But if you have intrinsic problems also in the health, those people are much more vulnerable. That is the reason. But I am very firm of the firm belief that these vaccines would be developed and this particular virus would be defeated for sure. And I'll tell you in a minute as to why. So you as a biotechnologist here or people who are working in the industry, you have tremendous opportunity. So take a regular flu toolkit, a simple one. There are various types. So take a simple one. Open that toolkit. And in that toolkit, just one area that senses, put these nanofibers that we made, or put graphene quantum dots, or for example, silver nanoparticles, or few other nanoparticles that are smaller. Then put the toolkits back. I'm very sure COVID-19 strain, which is a viral strain, which is a pathogen, which will be detected much faster. Why? Because these particles or these surfaces have a very high specific surface area because they are nano. As they're very high specific surface area, even the least quantity of this strain would be able to be very fast. The next goal would be, if there is a silver particles there, silver particles can also alter RNA. So if you alter the RNA of this vaccine, of this strain, now the strain is confused because it's not being able to get the right signals. So RNA is the one that carries the order, like the faculty gives order to their students in a very principled way, student follows those. Cell instructions cell gets from the RNA. So RNA gives the instructions. Now, if you confuse that RNA by putting the small particles, RNA binds to them, then you can also kill this pathogen as well, not just detection. So future world is the world for biomedical engineers or bioengineers, if you will. Biotechnologists, biochemists, bioengineers, pharmaceutical people, pharmaceutical scientists, or virologists. They're the one. They're going to get tremendous amount of boost for this. Do we want this pandemic? Absolutely not. But I feel the challenging creative people are those who don't ride the wave of time, but those who challenge time. And we can. As you see some of these works, I'm always pained that I'm home. We're not being able to go to offices, but I'm constantly thinking. We proposed to National Science Foundation about a proposal to find, is there any connection between brain and COVID-19? So from smell, from taste, from senses, we can detect that. So we proposed, we call RECODE. So RECODE is, reprogramming of cortical organoids development by engineering. So on the RECODE, we proposed using COVID-19. So many of you can do. So there's a tremendous amount of opportunity right now for bioengineers. So bioinstrumentation, biomechanics, biomaterials, system physiology, clinical engineering, rehabilitation engineering, all are biomedical engineering, biorobotics, bioinformatics, bioelectronics, biofluidics, biomemes, biosignaling, bionics, all these combinations that you see here, our program tries to integrate courses along these lines into our biomedical engineering department that I founded. So we bring in expertise of various people to make it a full-fledged program. And somebody working in a robotics field can do a lot of robotic surgery or robotic guidance onto certain biological problems. So you can imagine machine learning, artificial intelligence. With the artificial intelligence coding, people have been able to predict the strain changes of these proteins faster. If there's a cancer hidden from an image, you won't see it. But if you reprocess the image by AI, you can make 5D image and find out the pixel density, and you can find pinpointedly the points of interest for these cancer cells that has come up. So by AI, artificial intelligence or machine learning, you can integrate that into a BME field also. For example, we have been working on benchmarking algorithm that can talk about how neurons talk to each other. So if there's a neural disorder or brain disorder, you can really still use AI in there, machine learning. It's such an important field. But for that to realize, people at the senior leadership has to be visionary. Professors has to think ahead of their time and advise students about their career. So in other words, many of you biotechnology students, be very proud of yourselves that you'll be getting jobs that don't exist yet, which means don't think that jobs are not there. Some of the jobs you will be creating for yourself. But in other words, if somebody works on bioimage processing, you can develop a software tools that will be sold to various universities or various companies who buy your tools. Now you are owner of a company, so you created that job. This field is such, it has existing jobs, but also the jobs that are for future. And I can show you some examples where our students have gone. This is a statistic from 2010 to 2013, but I can say you up to 2018. Many of our students are in Pfizer. They're in Johnson & Johnson. They're in Medtronic, one of the largest biomedical electronics company. Mayo Clinic, Abbott Corporation, Illumina, Bristol Myers Squibb, CVS Health, Cadence, Unilever, Canon, you name it. Honeywell, glaucoma. So glaucoma is a company that made optical lenses so that glaucoma disease could be overcome. Various biomedical engineers of ours are working there, and it's a Harvard-based company. Various of our students have gone to work as an engineer in Harvard's hospitals. Harvard is a combination of hospitals, and there are many local area companies that I don't name here. I only name the big companies. 3 Psi Technology right side, if you look at Nashville, Tennessee. Then Rothal Healthcare, New York, Hartford Hospitals, Instant Labs, Beth Israel Hospital Center, Boston, Medtronic, George Mason University. There are various places our students have been hired. This is just a few examples. Or in other words, whoever is graduated with a master's degree, they all have jobs. So job is not a problem. Pathway is something that you have to design, meaning study well, get good grades, but more than that, get hands-on training. Try to solve problems by doing great master's thesis work. Develop something, be creative. Then company will attract you like easy. The job is an afterthought at that point. So, and so many interdisciplinary things that I showed you. I don't want to show all this. So this is an image. You can see how interdisciplinary this field is. Molecular dynamic simulation, validation of sensors, electrochemistry, physicists all work together in this field. This I already showed you. You can see here, we are developing these electrode containing patterns, EEG devices. We are modifying those EEG devices. We are also changing the ECG electrodes ourselves. These are very cutting edge biomedical research that you could do based on which company will hire you. Look at this bacteria. It is helping us validating the logic circuit. It is helping digital logic circuits design. It is helping us to track bacterial movement. So various things, those who have developed those, they're all in a very good biomedical companies or pharmaceutical companies. So with this, I'll tell that our university is a very good university. It has 6,000 to 8,000 students. Students-teacher ratio is very small. Hence, students can get access. I'll again take a few moments that Sindhu is a student from your college to ours. She's doing amazing under my direct supervision. I'm very sure after graduation she'll get a job. Sindhu came from microbiology, zoology, and chemistry, MGC from you, and she worked under Dr. Grace Paul. And she is very much indebted to all of you for studying in your college. And with this, I'll show you a few pictures where our students are, what they're doing. With this, I'll end. And these are all the companies that hire our students. beyond the companies that I listed. With this, I'll end. Thank you very much. And I'm very, very grateful for all of you to, particularly Dr. Padmalatha and your biotechnology department, your principal, your senior leadership for allowing me to do this presentation. Thank you very much.
Thank you very much, sir. Indeed, it was an excellent presentation. We have got lots of feedbacks, which is having good comments on that. And we have few questions also.
Sure, sure.
Can you please answer? I'll read one by one.
Yeah.
Kiran Kumar has given, using which equipment these cells are studied? Using which equipment?
Yeah, so that's the question. So it's not one equipment, it's a series of equipments we have used. For example, if you want to study, attachment of cells to produce the scaffold, you use electrospinning apparatus. Then to study cell growth, there are various way of doing staining, immunostaining, and you see how much they are doing. Then you can count cells, cell cytometer. Yes. Then you also have, when more and more cell comes, more and more endothelial network forms. So you can measure conductivity change by 4-point probe or by impedance. So that's an engineering measurement. Then you can do confocal microscopy. You can do scanning electron microscopy. You can also do transmission electron microscopy. So there are various technique: imaging, analysis, histology, immunoassays, and also you can do single cell transcriptomic analysis. So using those, you do deeper understanding of those processes. So if you don't use the apparatus, then you don't get details.
Yeah. Kiran Kumar, I think you've got the answer for this. And next question is, recently some nanoprobe based diagnoses are being developed for COVID-19. Will they be superior to ELISA or RT-PCR?
So that's a good question you asked. Whether they'll be superior or not, I don't know, but I can tell you we can find the scientific merits of those. Those nanoprobes are of extremely small size. The functional parts of the nanoprobe is nano, not the entire probe, right?
Exactly.
Now, these strains that COVID-19 has, this is a protein, and this protein folds in femtoseconds. Now, think about a nanoprobe that has 10 to the minus 9 meter, area over volume. So that means what it can do is immediately it can attach these strains before they folds, or it can alter the folding adsorption processes. So strain change would be lower or slower. When that is slower, meaning your action would be also slower. So various groups are working on nanoprobes. Many of them are not also very successful because the interface formation is key. But nanoprobe is particularly for detecting and sensing these pathogens. And RT-PCR and ELISA also have their own place. In their own function, they're very well. So it's not about better, but it's a complementary. You can say it's not competitive, but it's a complementary.
Okay, sir. I think Geetanjali ma'am, you got the answer for that. And the next question is asked by Haimavati. What role does chemistry in nanotechnology play in biomedical engineering?
That's a very good question, actually. I personally feel that without chemistry, none of these good stuffs could be developed. And let me tell you this, why I'm saying this. Myself, I have undergraduate in chemistry, by the way.
Okay.
My PhD is in material science and engineering. My postdoc is from one of the top-notch university in this country, Rice University, on nanotechnology. Okay. And I have another postdoc from Massachusetts, is on bioengineering. So I have seen these fields in my own life very well. So when you grow nanofabricated devices, there is a step called lithography. And the lithography is about chemistry by light or by X-ray or by chemical treatments. So chemistry is very important from synthesis, also altering or tailoring the surfaces, attaching or detaching single molecules. So chemistry is an integrated part of nanotechnology. It has to.
Okay, thank you. The next question is, what are synthetic nanoparticles? Asked by Sujata.
So what is the question? What are the synthetic nanomaterials, right?
Yeah. What are the synthetic nanoparticles?
Okay. Synthetic nanoparticles meaning the particles that are not in nature or human body, but we are making it. So one of the synthetic nanoparticles would be, for example, certain nanopolymers. They could be a vesicle structure, just like blood vessels. So it can basically understand those vessels like geometry, morphology, and its role. It's one side. Second is synthetic nanoparticles could be PLA, PLGA, polylactic acid, PCL, polycaprolactone, polylactic acid, polyglycolic acid. They are all biodegradable polymers, but they're also available as nanoparticles. So they are synthetic nanoparticles.
Okay.
Similarly, you can make various biological synthetic molecules. They are not natural molecules, but they're synthetic, but they behave like natural
Okay, sir. Question is, nanotech probes more effective and sensitive than PCR?
Repeat the first part of the question again, because that voice was getting.
How nanotech probes more effective and sensitive than PCR?
So PCR is for multiplexing, right? Polymerase chain reactions. So
Yes.
PCR is also undergone changes, RT-PCR, by depending on changing the surfaces of the machines and all. You're talking about nanotechnology or nanoprobe.
Nanoprobes.
So nanoprobes are used for various purposes, not just for PCR. So I would say that's a multifunctional probe. PCR has its own role and place. So again, I'm saying it's not better, but PCR technology can be improved by even nanoprobes. For example, cell sequencing is done by various, high sec. It's called high resolution sequencing, right? They have the chambers, and they have patterns, and they have channels, right? The moment you make nano channels, your efficiency is going up, and you're putting many such channels. So nanotechnology is something that is to improve these existing technology and also bring in new ideas for application field.
Okay. Thank you, sir. Thank you so much. And one question. How to control, with tissue engineering method, heart and other organs can be developed, but how to control the immune response in vivo conditions?
That's a very good question you asked. So that is the reason, when I'm saying these, I honestly mean that I don't mean that we have solved everything in the world. What I mean is, with this nanotechnology or tissue engineering, you can understand various complex processes. So when you make these materials, people have studied how biocompatible they are. Are there any allergic reactions from these surfaces? When they go to the physiological environment in a human body, do they affect them badly? By knowing tissue engineering, you can see various implants also have been designed now, right? So to answer to your question, it is not easy to do all the functions in vivo. It might need replacement, but at the same time, now with the modern technology, the moment we put something inside the body, we're also getting feedback in every minute. So there's a new area called wireless monitoring of implants in the body. Earlier, people say your implant is not working, so we'll recall it. So after 6 months, you go to doctor again and then take it out and put a new implant. Now patients are saying, no, I want to know every minute what it is doing in my body. So we have a sensors wirelessly giving out the signal. So the idea would be someday, indeed, we'd be able to go very close to in vivo, as much function as we could. That's the aim. But yes, in vivo and in vitro are two different things. So there are challenges. I agree with your question.
It was an excellent answer, sir. Thank you for that. And two more questions.
Sure. I'm open to take as many questions. No problem.
Okay, sir. Can zoology graduate go for this biomedical engineering after their PG?
Can zoology, right?
Zoology. Yeah.
I think absolutely. I think Sindhu was one of the students from that program. Microbiology, zoology, and biochemistry, whatever the field, you can come. And let's say if you don't know a lot of area, we still offer core courses, so we prepare you for that. Then you do core courses, you are now trained onto the core area of biomedical engineering. For example, molecular cellular bioengineering, tissue engineering, biosensor electronics nanotechnology, then synthetic bioengineering, and then let's say I should say neural engineering. So if you take 1 or 2 courses each, you are already being trained. So you can absolutely come from any field, so long they are science and engineering field.
Okay. Okay, it's excellent, sir. I think more of zoology students will join in that.
Yeah,
Thank you.
absolutely.
Yeah. And some nanoparticles which are used in soil organisms. Can you please about that? It's asked
In
by
you mean in which organisms in the soil?
soil organisms. Yes.
If I know the questions, I mean the purpose, then I can answer much more precisely probably. But nanoparticles are also being used in agriculture, for example.
Oh,
So in soil organism, if I know there is a group at MIT who is very known to me, Michael Strano. He's using nanotube sensors that is giving feedback to the leaves, tree leaves, how their breathing patterns, how their photosynthesis is happening. Similarly, you can also mix these some of the particles to activate some of the fertilizers. So to bring hybridized better crops, you can use nanoparticles. At the same time, you can also kill insecticides.
okay.
Those chemicals, harmful chemicals, you can nullify. So depending on applications, they're being used in soil conservations and various other agricultural benefits. They're being used, but in a very specific way. So not that all problems are being tackled.
Okay. Okay. Thank you for that. And the next question is, what are the present strategies to develop the vaccine for COVID-19?
Yeah, that I can answer, more or less in a very honest manner. You can see there are many people claiming every morning that they have got the vaccine. That's not the case. I, for example, myself know one company. I have several friends there that is Cambridge-based Moderna. Moderna is the one that is very close of developing the vaccines. That is also developing antibody. But many of you should know here. You should be very informed, being a biotechnology student or in the community. Antibody is being developed in the body. It also doesn't mean anything. So antibody developed doesn't mean that that's the cure. So you have to have the vaccine. That vaccine will develop antibody that has ability to interact with this particular protein strain and hence nullify it with the vaccine. So all vaccines are creating also the false antibody. So Moderna and remdesivir, and I know that Oxford didn't, claimed but didn't pan out. Even Moderna vaccines, which is very close, but still they have issues. But I personally believe there are a few other companies like Novartis and Pfizer is also in the business. Even Boehringer Ingelheim, a pharmaceutical company, they're also pretty much working very rapidly. But my feelings by 2021, middle or end, hopefully we'll see some good solution for this.
Okay. Thank you
But
for
I
that.
but I would suggest, ginger, garlic are extremely efficient at this point because they're very rich in antioxidant. And whenever there's an antioxidant, it has a pathway as a free radical to attack this, strain site. At least it can lower. So hopefully, but I can update you more as I see the progress here on the modern or the vaccine.
Exactly. Our Indian herbs are very much useful.
Yeah,
Yeah.
yeah. And I see some of the Indian company also or labs, they're doing some amazing work from this antibody development and then sequestering the antibody and finding which is the perfect combination. I
Yes.
don't know the name, but I believe some of the amazing work is being done in India, and I'm very positive about it.
Most of the vaccine companies are in Hyderabad.
Oh yeah. I used to go to Hyderabad actually sometimes twice a year, sometimes once
Okay.
a year
Yes,
before
sir.
before this virus shut down all the airports.
Yes. So one question
Yeah,
asked by Zainab. I need to ask about my future opportunities with my microbiology stream.
red hot. It is one of the best field. If you are a microbiologist, I have several students who are microbiologists in my biomedical engineering program. Several UB undergraduate biology, they are coming into BME program also, our own students. Sometimes I don't feel very good because biology colleagues are right there next to us. Their students are coming to our program. But the moment you are an engineer, you're open to devices, open to pathway determination, open to sensing industries, and then also utilize your own microbiological pathway. Microbiology is the key. And you can see some of my work that I showed you, right? The bacteria that is done in microbiology way, but they're doing amazing engineering. So microbiology is a great field. And I, as a researcher or as a faculty, I'll not say any field is bad. If you can utilize the field with a real vision and good creativity, all are great, but microbiology is really, really a very sought after field.
That's true, sir.
Yeah, every field has its unique strength. And
Yes.
bioengineering is a much more interdisciplinary that covers all these aspects.
Yes, sir. And the last one, any funding from your university for our Indian students?
Yeah. So all graduate students get some sort of scholarship. What I'll do is so that I don't misspeak, I will ask our graduate admission office to tell you exactly the numbers and all. It depends on your GPA and some results. They give 5,000, sometimes 8,000, depending on that. Yes, scholarships are available. Yeah, and then that is one. That is your admission time scholarship. But also when you come, there are on-campus opportunities. Even though you don't get scholarship, some of you, but after coming here, you can arrange for scholarship by talking to professors who need TAs. Then also on campus other activity, you can do some voluntary, you can do some support work for some other professors, and they give you some money, and then you cover your tuitions and come back to your own department and do stuff. So there are a lot of opportunities exist, officially speaking. So,
Okay.
but
Thank you so much, sir.
exact number I can email you once I talk to Erica Castillo, who is our admission counselor for India.
Okay. Thank you so much, sir. I hope more students of ours will be flooding to your university later, the
Oh,
next
no
academic.
problem. Yeah. Please
Yeah.
ask them to individually email me to pack their questions, make a list and send it to me. Any scientific questions I'll be able to answer. Admission related questions, GPA, etc. What is needed, that I can tell also. But how much is the scholarship? What is the financial aid and all that? Admission office knows more, and they are authorized to speak more on that. So I don't want to misspeak on this.
Okay. So kind of you, sir. It was
No,
really
thank
from my
you.
personal. Yeah.
Thank you to you. I mean, you are the one who invited me, and I got connected to your school, and hopefully
Yeah.
someday I'll visit your place.
Yeah, sir. Welcome, sir. And we learn more from you, like electrospinning, about the heart cell video. It was amazing. It was wonderful. And insights on the tissue engineering. And you created research interest to the students also,
Yeah.
and little knowledge about quantum dots too. Thank you
So
so much.
I forgot to mention that in our program, thesis is a compulsory. So they will develop their own thesis. So they'll be working on some projects, the hands-on. So thank you. Yeah.
Thank you so much, sir. Now I request our faculty, Dr. Anjana Shukla, to give vote of thanks. Thank you.
Thank you.
Anjana Shukla, you can start.
Yeah. Okay. Good morning, sir, and good morning, everyone. The best and beautiful things in the world. Can you hear me?
Yeah.
Yeah. Okay. Thank
Please,
you. Okay.
please.
The best and beautiful things in the world cannot be seen or even touched. They must be felt by heart. Thank you is one such prayer among them. On behalf of St. Pius X Degree and PG College for Women and Department of Botany and Biotechnology, I, Dr. Anjana Shukla, take this opportunity to propose vote of thanks. I would like to thank our today's speaker, Dr. K. Patra sir, for giving an excellent coverage to nanobiomaterials in biomedical field. And sir enlightened mostly our students about engineering, career and education opportunity. Since it is a multidisciplinary approach in which all life sciences and physical sciences students get drastic change in their career in science and technology. I'm sure the student will have a lot to take away from this program. Sir, we are really enlightened with your knowledge and presence. We have been fortunate to be assisted by our principal, Reverend Sister Velangini. You have been a constant support and you have given us a lot of encouragement. Thank you, sister. This event would not have been successful without the help of Sandhya Sri ma'am. Thank you, ma'am. I would like to thank our HOD, Dr. P. Padmalata, and my colleague, Mrs. Suganmala, who have made this webinar a grand success, as we are getting positive feedback from all participants. Finally, I would like to thank my wonderful participants to showing their interest in their webinar and their active participations. Thank you so much for your cooperation. Thank you all of you for making this webinar grand success with your contribution. Thank you. Thank you, sir.
Thank you, thank you. In fact,
And
let me tell you,
yeah, sir.
I like your university so much, and the way you did it. So it's all credit goes to many of you. Please.
Okay, thank you, sir. Thank you
Thank
so
you so
much.
much, sir. Thank
Thank
you.
you so much, sir.
We
And
And
got a lot of positive feedback, sir, from the participants in the
yeah,
chat box. You
and
can
one
go
thing,
through that also.
yeah, one thing I forgot to tell that if any student wants to know more about heart cells, you see right there the publications mentioned, the name, the page, and volume. If they dig out the paper, they will have all the movies in there, so they can see various things in
okay,
details, or I can send them if they're interested. Yeah.
okay, sir.
Okay, sir. Sure, we'll do that.
Thank you,
All
sir.
right.
And one more announcement for participants. If the feedback form is not opening, don't be panic. It will open. Slowly it will be open. Until 5 o'clock time is there, you can fill the feedback form. So please do it.
Thank you once again. Thanks everybody.
Thank you.
Dr. Mala,
Thank
Dr.
you, sir.
Padmalata, Dr. Sukla, and everybody. I'll again take the name of Sindhu and Dr. Grace as well because they provided me the
Yeah.
connections. They're very
Yeah.
early enrolled for this. And
Yes.
any way I can help your students, any way we can establish collaboration, please feel free. I'm as a team, and we believe in transparency and all, and we'll be able to go up to any extent to help these kids. And no problem at all.
Thank you for
sir.
your kind
Thank you
words,
for your
sir.
kind words.
Thank you very much.
Thank you,
Have a
sir.
wonderful
And
day. I think for me it is 1:45 a.m. now, early morning.
yeah.
So
Sorry.
thank you.
Okay.
Thank you.
Okay, sir.
I'm
Thank
signing
you.
off. Okay.
Yeah,
All
thank
right.
you so much, sir.
All right.